everyone. I am Dr. Charu Dogra Rawat, Professor at Ramjas College, University of Delhi, and we have been discussing in biotechnology about the production of cloned and transgenic animals. Today we are discussing about micro-injection method of the generation of transgenic animals. Now we superovulate the females and then we breed them or mate them with normal males and we get the fertilized eggs. And in these fertilized eggs, we introduce our gene of interest to create the transgenic mice. Simultaneously, we breed a foster mother, of mice that we are going to, a female mice that we are going to use as a foster mother for the implantation of the fertilized egg carrying our gene of interest, and we breed it with a vasectomized mice, which is not producing the sperm so as to create the pseudo pregnancy. Now, how the superovulation is done and how these fertilized eggs are harvested has been discussed in our previous session. Now, to micro manipulate these fertilized eggs and introduced or micro inject our gene of interest, such kind of micro manipulators are used. These micro manipulators contain microscopes and they have got the platform where this kind of micro injection can be done. Now, as you can see in this platform, this fertilized eggs are taken on the slide, the medium is added to it, and then a layer of oiling is also done. 2 drops are just put at the end so that they form a film around this fertilized eggs containing media. This is basically to prevent the evaporation of the media as well as to so that the zygotes are not dehydrated or the zona pellucida is not separated from these eggs. So, this is how it looks like, and this is how on the platform the eggs are basically put here. Then as you can see in these photographs, the suction pipette or this glass pipette basically holds the fertilized egg in place. Usually it does at the point where the polar bodies are there, and so that these female and male pronuclei are aligned behind it. Now, male pronucleus is usually larger, it can be identified separately, and this is the female pronuclei. And then from the other side a micro injection is inserted, and this micro injection is carrying our gene of interest and it injects that transgene into the male pronuclei. Now, male pronuclei as shown in the inset is basically swelled up, it swells, and as you can see that the micro injection is put through. So, it is kept in place by use of this glass pipette, and then micro injection of the transgene is done directly into the pronuclei as shown in these photographs. Now, this male pronuclei then generally swells up and can be identified that the micro injection has taken place. After this micro injection step, the reimplantation of the injected zygotes is done. So, 25 to 40 eggs are implanted microsurgically into a foster mother that has been made pseudo pregnant by being mated to a vasectomized male. So, they are having the same cycle, they are in the same cycle, and they have been, as I said in our previous session, that how the vasectomized male are produced, and the breeding is done simultaneously with the superovulated female and a normal male, so that you get a pseudo pregnant mother, foster mother, which is at the same stage as our superovulated mother. These foster mothers will deliver the pups from the inoculated eggs about 3 weeks after the implantation. So, this is how the reimplantation is done. The zygotes, a number of zygotes, just like the pearl arrangement, just like small pearls are taken in pipettes, and then even the some air bubbles are also kept so that they can be visualized, the transfer can be visualized. And as you can see in the mice, the incision is made here, and by the fat pad one can identify the ovary, and then it is pulled out. So, you can see there is ovary, there is oviduct, and there is then the uterus, and basically it is identified by this fat pad, and this transparent is the oviduct, and then simply these fertilized eggs microsurgically basically they are introduced into the oviducts. into the uterus. So, they are basically implanted in here, and then they are left to basically develop. So, this is the, and then it is sewed and everything. So, surgery is done, and this is how the fertilized egg carrying our gene of interest. So, the transgene is implanted or re-implanted into the foster mother. So, that is how it is done. The last step for the transgenic mice, to identify the transgenic mice, is genotyping. Now it is going to produce a series of pups. As I said, about 3 weeks after implantation, it will deliver pups. Now, to identify whether these pups are carrying our transgene or not, genotyping is done. The genomic DNA is extracted from the tail biopsies of the 3-week-old mice. It is done by hot digestion. So, in hot digestion, basically the tail mice is warmed, or it is basically kept in a buffer, lysis buffer, where it is made hot by putting at 95 degrees Celsius for some time, and then it is put on ice, and it is neutralized, and then centrifuged so as to get the supernatant, and in that supernatant we can check for if the transgene is present or not. This presence of the transgene in the genomic DNA extracted from the pups of the foster mothers can be done by southern blot hybridization or polymerase chain amplification, PCR amplification of the transgene. Now, both these techniques we have discussed in detail in our advanced biotechnology sessions, where we talked about various techniques, advanced techniques. So, either they can be hybridized by using the probes which are specific for the transgene that have been introduced, or we do the PCR amplification, in which we design the primers which is specific for the transgene that has been introduced. So, then genotyping is done, and the pup carrying our transgene, which are then called as the founder organisms, are basically identified. These founder mice, a transgenic mouse, can be mated to another mouse to determine if the transgene is in the germline. So, first is we identify these founder organisms, but then these founder organisms are bred with other mouse, so that to understand if the transgene is inherited or not. So, if it is in the germline, we require the one which is in the germline, so as to create a transgenic line. We just do not need one organism so that it dies and the transgene is lost. We want to introduce it into the germline and create a transgenic line which will purely breed with the presence of the transgene. So, we basically determine if the transgene in the germline is present in the germline by mating it with another mouse, and then these founder animals subsequently progenies can be bred with each other to form the pure or the homozygous transgenic lines. So, these homozygous transgenic lines are then obtained and maintained, and then the expression of the transgene or the function of the transgene is studied in the mice. We will talk about the application later. We are just talking about the methodology where how the transgenic mice are produced. But we will talk about the application of these transgenic mice subsequently when we talk in detail about the application of the recombinant DNA, or application of biotechnology, or application of these transgenic mice as well. So, this is basically the timing of the entire process. So, you can see that we start with the cloning of the gene of interest, and then after cloning of the gene of interest, we superovulate the females, we cause the microinjection, then we retrieve the fertilized eggs and reimplant it into the foster mothers. The zygote formation is there, and then the breeding is done so as to create the founder animal. Basically these mechanisms, they do not take much time, and they can be done one after the other. But obviously when you get the founder animal, then initially the superovulation takes time, and later the transgenic mice breeding, getting the founder animals and producing the progenies to get the lines, it takes around 2 to 3 months for the pups to be there. and then the genotyping again does not take much time if it is done even by either by southern blot hybridization or by PCR amplification. So this is the timeline that how this transgenic mice production is done and how much time does it take. So we take one example of the genotype of generation of a tau transgenic mice. So we'll just take as an example so that the methodology is further understood by you. So tau is basically a microtubule associated protein of a mature neuron, and it contains two major domains. So we'll talk about one protein which is a tau protein, and this tau protein is a microtubule associated protein predominantly in the mature neurons. So it is found in the brain in the mature neurons, and you have this protein. So we want to clone the gene for this protein and we want to express the gene of this protein in the mice. So we want to create a generate a transgenic mice containing not the complete protein but one of the domains of the protein. Now this contains two domains, projection and a microtubule binding domain. Now to dissect the role of these domains, this is what we want to basically do, that we want to separate these two domains and then study what their functions are. So to dissect the role of these domains, the projection domain of this is expressed under the control of a promoter, for example mThi 1.2 promoter, and this has been represented as delta tau. So this is containing, so delta tau basically means that it is containing only one of the domains, and then it is expressed or cloned under the action of this particular promoter. So what happens is, when we want the expression of certain transgene, then we do clone it with certain regulatory elements as well, so that when it is introduced into mice, it does contain its promoter which will express that particular gene. So in this case the delta tau is cloned with this mThi 1.2 promoter, and this particular stretch of DNA, then recombinant DNA, is then introduced into the eggs. Now after linearization with NOT1 and PVU1 and removal of the vector sequences, the DNA is injected into fertilized oocytes. Now 39 pups. So this is a specific example. So in this specific example, what they did was they cloned this tau gene, the delta tau gene, under this promoter, and then they cleave it with two restriction enzymes, which is NOT1 and PVU1, to get the DNA that needs to be injected, and that it does not contain the vector sequences. So I told you why we cloned it into the vector. We cloned it into the vector so as to get the more number of copies of the transgene. So after and to check whether this transgene is expressed or not, or what is the amount or the quantity that it is expressed. So after that we take this transgene, we digest it, and we need to inject only the DNA without any vector sequences. So after this injection, around 39 pups, 20 female and 19 male were born 20 days after reimplantation, and then they were genotyped by PCR upon weaning at the postnatal day 21. So after 21 days you get 39 pups, 20 female and 19 male, and when they were analyzed by genotyping as shown in this particular figure, you can see this is the structure of the transgene. So you have got the delta tau gene, and along with it you have got its promoter. So this stretch was introduced into the mice, into the same mechanism, the super ovulated eggs. By micro injection this was inserted, and then the transgenic pups were obtained by reimplanting the zygotes, the carrier transgenes, into the foster mother. And after 21 days you do the genotyping by taking the tail biopsies and amplifying it with the primers that causes the amplification of this delta tau genes, which has been designed to amplify this delta tau gene. And as you can see, in the 20 female and in the 19 males, around 6 females and 1, 2, 3, 4, 5, 5 males gave positive amplification. So when you amplified the transgene from the genomic DNA extracted from the tail biopsies of the 21-day progenies, the pups that were there, then you obtain that 6 gave a positive result in the females and the 5 gave the positive result in the males. So these were basically the transgenic mice 6 and 4. 6 and 4, this is the positive. So 6 and 4 pups, basically there are 10 number of pups that need to be identified. So 6 females and 4 males were typed transgenic positive. So we will analyze now these lines further. So for the further thing, 7 of the 10 breeding pairs gave offsprings. So 7 of the 10 breeding pairs. So when we obtained these 10 breeding pairs, we further mated them, and then after mating we got around 7 of the pups, they further gave the offsprings. So we initially got amplification in 10 pups, then we got 7 pups which basically gave rise to the offspring that still carried the transgenics. Now transgenic animals from the F1 generation were again mated with the mice to establish these 7 individual delta tau transgenic strains. So this is also telling you that how does that occur. The cloning part is done in vitro, the superovulation part, the microinjection part, everything has been done. But then when you identify the transgenics, which you do by any DNA manipulation method, but then you have to breed them again and again so as to get the transgenic lines where the transgene has been established. So in this case, when you breed them with 7 mice, you obtain, you identify that out of these 7 strains, 2 of them produce the progeny which did not give amplification with transgenic again. So all you get was, you analyzed a series of pups of the 2 of these transgenic mice that were obtained, but they always give wild type amplification. They did not give transgenic amplification. So those 2 lines were not analyzed further, and then one of the lines out of these 7 lines did produce transgenic mice, but their number was very low. So the litter size was very low. So they were producing only 3 to 4 progenies per litter, and even they was taking long time, long gestation periods. So regular gestation period, but they are taking much longer gestation periods. So again, that is not very useful for us because we want that the transgenics are produced at least in a larger number, and the gestation period is also small. So they left those 3 transgenic mice that were obtained and analyzed rest of the 4 for the expression of the transgenic tau protein. The PCR amplification shows us that the transgenic transgene is present in these lines. The DNA sequence is present in these lines, but we just don't want the presence of the DNA, we also want its expression. So the expression of transgenic tau protein were analyzed, and you can remember when we discussed about the techniques that to analyze the expression of a gene or the formation of a protein, Western blotting is done. This Western blotting for the expression of this transgenic tau protein is done where a human tau specific antibody of the brain extracts was utilized. So in Western blotting, you remember that we isolate the protein, transfer it into a nitrocellulose or a nylon membrane, and then hybridize it with the antibodies which are specific for these proteins. So there is a primary antibody, there is a secondary antibody, and then we do the detection part where the signal can be detected and the expression can be analyzed. So in the same way, the Western blotting was done to identify or study the expression of the transgenic tau protein in the 4 transgenic lines that were obtained, and the selection was done by using a human tau specific antibody of the brain extracts. So in here you can see that in the top panel there is Western blotting result. So the 4 transgenic lines that were produced did show the expression of this tau protein. However, you can see that the expression is variable. 2 of the lines are producing a large amount of transgene. However, 2 of the lines are producing less amount of transgene. So, because it is a random insertion into the genomic DNA, it may be introduced into different regions where there can be that the expression can be hindered. So, we have got the variable expression types in these things. And as you can see in this microscopy, there is expression of, in the hippocampus, the expression of human tau was limited to the cells of the neuronal types CA1 and CA2. So you can see this red one is basically showing the expression of the transgene, and the blue is showing the nucleus. It has been stained by DAP1, DAPI. So this is how the transgenics for delta tau is obtained, and in here they have obtained two, high and low. So two low and two high expressing delta tau protein. So I hope the creation of transgenic mice is in a way better clear to you, and this has been done by the microinjection method. There are certain limitations to that. For example, the success of implantation and giving birth to the offspring is much lower. As I said, only 5% or fewer of the treated eggs become transgenic progeny. So you start with a large number of eggs, but the efficiency of the production of transgenic mice is 5%. This shows, in different animals the limitation is there. And this can be due to the transgenic related factors, the method related factors, or the host related factors. In transgenic related factors, it depends upon what kind of promoter elements you are using. If you want to create these transgenic into specific tissue types, you need to use the promoters which are expressed in those tissue types. It depends upon the site of integration, because it may be integrated as it integrates randomly, that it may be integrated at certain regions where the expression is hindered. Again, the copy number, in one way, more the number of transgenes will favor more the production of protein, but in some other way, it can hinder as well the generation of the litter or the sustenance of the litter as well. Method related factors, there is buffer compensation, DNA concentration, site of microinjection, and the type of DNA. So, and in the host related factors, the particular reproductive biology, the sex stage, the egg morphology, the litter size, the site of production, all these factors are going to influence the production of the transgenic mice. As I said, there are certain specific promoters that you need to use if you want to express the transgenic in particular cell lines, for mammary gland, in liver, or in skin, or in muscle, erythroid cells, so a number of proteins basically can be used so as to express in certain tissue types. As I said, randomly, and copy number can be inserted. This shows how the method related factors, the DNA concentration will depend. A certain amount will not produce any transgene, but if you use a certain amount, certain efficiency would be there. The form of DNA, you are linear. As I said, linear, they are going to produce more transgenic. Probably in supercoiled, there are no transgenics that would be formed, or less number of transgenic. Buffer composition, or the site of entry, if you enter it into the nucleus, you may inject it, but it should not introduced into the male pronucleus at all. Then also the transgenics are not formed. And this is the last one where where I just show the reproductive events in different mammals, which are basically the host related factors. So you can see that varies. This is a normal number of eggs they produce. This is a superovulated egg, the gestation period, etc. So one has to keep in mind these host related factors as well to create the transgenic mice, which are so that we can have an established cell line that we can use and employ in our subsequent applications. So that is all about the transgenic generation of transgenic mice by using microinjection method. In our subsequent sessions, we will take up the other different methods to create the transgenic mice. Thank you.